So we're moving on from external living things that can get at us and make us sick, and we're going to go to stuff in us. We will go to stuff from the outside that can make us sick before we're done with this section, but right now we're going into genetics. So a gene, remember, is a DNA code, and from that code you make a protein, and the protein does something. It may be a main player in producing some sort of trait. It's very often a contributor to some sort of trait. And every one of our codes to make that protein isn't the same from person to person, even since we have one of those codes we got from our mother, one of those codes we got from our father, they may not be the same code exactly. And then it's a question of, so if the alleles are different, let me just make sure that's reading that. If the alleles are different, then are they having an effect on the coded protein? Now, most alleles are technically different sequences, but the protein that you're making is functionally, for what it does, it's the same protein. So we all carry, some variance in us. Even from one cell in us to another, we carry some alleles. But it does become a big deal if you get an allele and the protein that it produces doesn't work or works very poorly. This is where dominant and recessive come in, which we'll talk about in the lab and talk about how those get inherited. But typically, a recessive allele is a code for a non-working or weakly working protein. And if you pair up a code for a protein that doesn't work with a code from the other parent, and it produces a protein that does work, then most of the time what the protein's doing gets done. So the fact that you have the bad allele, the bad protein allele, it kind of gets hidden by having a good one. Your body kind of compensates. It makes a little bit more of that protein to get done what needs to get done. And that's where the dominant recessive comes from. The presence of the working protein can hide that you have a non-working code. You only wind up in trouble when you wind up with two of them together, two alleles that make a non-working protein. So most genetic diseases are recessive. And in most cases, they're an allele for a protein, but the protein doesn't do what it's supposed to do. Now, most genes, there's not just one gene to make a trait happen. For most traits, there's a bunch of different enzymes, there's a bunch of different proteins, there's a bunch of different things that all together produce that particular effect. I mean, even the color of your hair, before mine turned gray, even the color of your hair there's a couple of genes contributing to that color. Even the color of your skin, there's, I think, 3 or 4 different genes contributing to that color. And then there's even more than that, because there's genes that make sure that the color gets where it's supposed to go, that it gets deposited in the right way. If you are an albino, you produce you have all the codes for color in your skin and color in your hair and color in your eyes, but one of the proteins that's critical in getting that stuff expressed where it's supposed to be expressed, you get 2 bad alleles. Neither copy makes a working protein, and so the color never gets into your skin, the color never gets into your hair, the color never gets into your eyes. And so that's why sometimes changing one protein in a feature that's produced by a bunch of them still can screw that feature up. there's some debate over just exactly how many genes humans have. It's probably somewhere between 12 and 15,000 genes. So you got a copy of each of those from your mother and a copy of each of those from your father. So you're talking about 24 to 30,000 codes to make all the different proteins that you use in all the different cells in your body. Now you started as a cell that had to be 2 cells and then 4 cells and then 8 cells and then 16 cells. And you also need to package your DNA, one set of codes to go on to your offspring when you're making a sperm cell or an egg cell. Imagine trying to do that with 24 to 30,000 separate pieces of DNA. All those codes being separate. You gotta make copies of them, and then if you're gonna make a new cell, you gotta make sure that all the copies that go one way and all the copies go the other way, so you get 24 to 30,000 copies. No, not gonna work. So what you find in living things is they reduce the number of pieces they have to copy and move around when they make cells to something that is not that likely to screw up. They stick a bunch of genes together on these units called chromosomes. And it really is kind of just a housekeeping thing. It gives you a manageable number of pieces to be able to move when you need to move them, to be able to make copies of and move around. Now humans have 46 of these. In 23 pairs. And that turns out to be a lot. A lot of animals have fewer than that. And the reason for that is, as the numbers go up, the odds of you having a problem making cells or distributing the chromosomes, you get more and more of a chance to screw it up. When I was developing as a fetus, and the cells were forming the top of my head, my scalp, somewhere up around here, a cell divided, and 47 chromosomes went that way, and 45 chromosomes went that way. How do I know? Because my hair is a combination, or when I had color, is I had brown alleles from my father and red alleles from my mother. And red very often is covered by brown because it's dark and it can hide red. A lot of times people have red highlights, but remember, it's a multiple gene trait. But what happened was both chromosomes carrying the brown went one way, leaving only the red chromosome in the cell that only had 45 chromosomes in it because it didn't have the brown. Both browns went that way. And over here, they were still covering the red up. That cell kept dividing with its 45 chromosomes. And it formed a little patch on the very top of my head that when my hair grew in, right, I was a fetus. When my hair grew in, I had a spot about the size of a quarter on the top of my head that was red, very red. Used to scare the hell out of barbers. Back when they were using scissors, they just snip, 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 and I'd know, they'd pause and you could almost hear them thinking, did I cut this kid? How's all this red, is this blood? Well, no, it was just a patch of red hair. It's, so even in that, you know, I am a victim of that 46 chromosomes don't always go where they're supposed to go. If you're making a sperm or an egg cell, you're supposed to get 23 chromosomes into the sperm cell to mix with 23 chromosomes from the egg cell. If you don't, if you send a sperm with an extra chromosome or a missing chromosome, or an egg cell with an extra missing chromosome onto form an embryo, gonna kill it most of the time. You go, well, what about Down syndrome? Down syndrome isn't just an extra chromosome, it's an extra number 21. And number 21 is one of the smallest ones. You have a handout that looks like this. This is an old picture of, let me get the stuff out from under there. An old picture of a bunch of our chromosomes. We have number 1 through number 22, and you'll notice that they get smaller. They get smaller and smaller and smaller and smaller. Here's the number 21. It's pretty much the smallest chromosome other than the Y that we have. And here's what happens if the sperm or the egg gets an extra chromosome. All of the codes on that chromosome that you're supposed to have 2 of to make proteins from, you have 3 of. So every time you need to make a protein while you're growing as an embryo, you get extra. And for all of these, somewhere very early in your development, you're gonna make too much of something that is going to kill you. So you're not gonna get born with an extra number 4. You're not gonna get born with an extra number 10. You might get born with an extra 19, but you are really messed up. Number 21 is Down syndrome. And you can actually see the effects of having extra codes because it affects the development of the face, it affects the development of the brain, it affects eventually it affects growth, it affects a bunch of things. By just having 3 copies for every protein on that chromosome when you need a protein from that chromosome. There is a condition, it's in one of your early handouts where they talk about DNA, where you may get an extra number 15 from your dad, but the only way it works is if you don't get a 15 from your mom. And there's something about the way that the 15s are programmed ahead of time, where if you get 2 from dad and none from mom, you get one syndrome. If you get 2 from mom and none from dad, you get another syndrome, because your development is expecting one of each, kind of counterbalance each other. One, you're tall and thin and have long, skinny face, and very often ears with points on them. They call it Elfin syndrome. There's a better name for it, but it's Elfin syndrome. And if you get the other combination, you tend to be small and squat, somewhat heavy. So you can see, it's very different. I've seen people who have this, so it's not terribly, terribly uncommon. But think about that. You need to match up a sperm that has one sort of chromosome problem in being made against an egg that made virtually the opposite chromosome problem for just one of these chromosomes, the number 15. And that does happen. So 46 chromosomes, we screw that up a lot. And as women get older and they're making egg cells, those egg cells were actually produced around the time that the woman was born. So the older you get, the more likely it is for that egg cell, when it goes to divide, to set the chromosomes up and get just one set, it's more likely that it's gonna mess that up. And so you're much more likely to have a Down syndrome baby if you're older, but you're also supposedly less likely to get pregnant. What really happens is that if you get pregnant with any of these others extra, it might show up on a pregnancy test very, very early. But by the time you go to the doctor and he runs the pregnancy test again, he'll go, you're not pregnant, because the embryo didn't last very long. It may have lasted just enough time to put out the signal that the pregnancy test looks for, but it isn't gonna get very far along before it just, something happens and it dies. So it's harder for you to have a baby when you get older, but it's not technically harder for you to get pregnant if getting pregnant means the sperm fertilizes the egg. Okay, you'll notice that there's labels on these showing where certain genes are. This is called linkage, is that there's no rhyme or reason to this except for the X chromosome, which is the only chromosome where all the genes are doing basically the same thing. It's taking and making make sure an embryo develops as a male. If it's not there, the embryo develops as a female. So it doesn't need a special female chromosome to do that, is that the X chromosome does a bunch of different things, not really connected with being female. But the Y chromosome, which we guys have, is that it has particular genes that turn on receptors and hormones and other kinds of things to make an embryo fetus develop as a male. But everything else is just a mishmash of a lot of different genes all put together. Like here, you have hemoglobin next to insulin, next to catalase, which is a very important metabolic enzyme, next to FSH, which is follicle stimulating hormone. Next to that processor I was talking about for color, that very often they label these things with labels from what diseases you get if the alleles don't produce working proteins, and there's albino. Here's ultraviolet repair and ABO blood group, just next to each other on the same chromosome, because you need to stick them together in order for housekeeping, but they don't have to be doing anything similar. But sometimes things do get passed on as packages because they're close to one another on the same chromosome. And we'll talk about that in the genetics lab. Now the thing I put up is old. This is why. And I know you can't read this. I can barely read this. But this is a more recent labeled copy, and they don't even have all the genes on here labeled. But these are all pointing at locations on the chromosome for these genes that do all of these various different things. And that's chromosome 9. That's one of the middle size chromosomes. And it's got lots and lots and lots of stuff. Over time, we identify more and more and more genes for where they are on the chromosome and what they're doing, what the proteins are doing. This one's a little smaller. That's the X chromosome. Male hormone insensitivity, placental steroid sulfurase deficiency. As I said, they tend to name these after diseases that you get. APOR syndrome, Fabry disease, things that you get hemophilia A, that if you get an allele on there for a non-working protein, well, this is the only copy you get. So dominant recessive is kind of out the window. If you get a copy for hemophilia on your, well, it's the X chromosome. Sorry, I thought it was a Y. Yeah, never mind. We'll talk about X and Y inheritance in the lab. I'm surprised there weren't a bunch more on there. Sorry. I knew I had this. That's what I thought I'd put up. Y chromosome. If you're male and you do get something on the X, again, it doesn't matter if it's dominant recessive. It's the only copy you get. Inability to express affection, selective hearing loss, lack of recall for dates. Oh, this is a joke. This is a joke. This is a cartoon. Channel flipping, inability to see or hear the obvious. Ability to remember and tell jokes. Sports page, the gene called BUD-E. Refusal to ask for directions, the anti-stop gene. Pre-adolescent fascination with arachnida and reptilia. Spitting. Okay. Sorry. I thought I actually had a Y chromosome now. I have too many of these things. So, when cells divide and they move the chromosomes where they're supposed to go, you start with 46, you make copies, you have 92, 46 go this way, 46 go that way. That's what happens in all of your cells. That's called mitosis. In production of a sperm cell, you want just 23 chromosomes in the sperm. You want just 23 chromosomes in the egg cell. And we will talk about that later on when we start talking about reproduction. So problems with genetics produce a lot of different things. We talked about dominant recessive and genetic diseases. I want to talk about mutations. A gene is a very, very, very long sequence of little bitty bits that are part of the code. It's a 3-to-1 code. 3 bits is translated into 1 amino acid in the protein. Now, you can get a mutation where one of the bits is miscopied. It either is substituted for another one, or it's left out, or an additional one is stuck in between. Those are all point mutations. They happen at one point on the sequence. And most of them don't do anything. You can swap in a different code or a different bit for another one and very often get the same amino acid you would have gotten, or one that hooks the chain in the protein together the same way. Or it happens in a part of the protein that isn't that associated with what the protein does. Right? Proteins are really big molecules. Very often they have dents in them that do certain things, or and if you have a mutation that changes an amino acid way the heck down here, it doesn't really matter. So we carry a lot of point mutations that are nothing. Doesn't mean it always happens that way. Sometimes you can swap one amino acid for another and change a significant thing about the protein. Sickle cell anemia changes the hemoglobin, the oxygen-carrying protein that's round, with or without oxygen on it, to a version that is round with oxygen on it, but is long and pointy without oxygen on it. And if the only code you have is for sickle cell, the only allele is sickle cell, then all of your red blood cells are full of that, and any kind of oxygen deprivation changes so many of the proteins that your cells change shape. They don't want to go through the little bitty blood vessels. But if you have one allele for sickle cell and one allele for normal hemoglobin, there's a mixture of that in the blood cells. And the blood cells have to get really depleted in oxygen for you to get enough pointy proteins to mess up the shape of the cells. Sickle cell is considered to be recessive. The thing about a mutation is anything that produces a change from what it's supposed to be of the DNA is a mutation. So a change of a bit, a movement of a piece from one part to another, a little bit of a chromosome missing, a whole chromosome extra, a whole set of chromosomes extra, which doesn't really happen in people because it would kill us. But you do find in other living things like plants. It's a mutation. So it's a very broad category. Anything that changes the DNA from what it's supposed to be to something else is a mutation. It could be really tiny or really big. When they discovered that that fruit flies had mutations, then they started to go a little nuts about it. And then they started to mess with them to see if they could cause mutations. And they found that you could expose them to certain chemicals and you would get more mutants. You could expose them to radiation and it would get more mutants. I'm looking at how long this video has gone, so I think I'm going to stop it here, and we're going to pick up causes of mutations in the next video, and the next video will be shorter.